So this last part of the chapter is about applications to life and medical physics. So with what we've learned, we now want to see how we can apply electric charges and electric fields, not particularly Gauss's law, to different fields in life and medical physics. So one thing that you've probably seen and heard a lot about before is electrophoresis. So that really takes advantage of charge separation through an electric field. Next, we're going to take a look at the electric field of a cell membrane. And so that is going to be more so modeling of lipid membranes. So with gel electrophoresis, what does it separate? It actually separates DNA or proteins. It's because DNA is negatively charged because of its phosphate group, and proteins can have positive and negative regions. So depending on whether you have a DNA or a protein, there's going to be different protocols for gel electrophoresis, but it's really just the same principle. A sample is placed in a block of gel, an electric current is applied, and it causes samples to move through the gel, negative towards positive. And the fragments are separated by size and also by charge. So the larger the fragment, the slower it's going to move along that current. So where is the E&M involved in this procedure? It's mainly the electric field that's generated through the gel that contains small pores. A negative charge at one end is going to push negatively charged molecules through the gel towards the positive end. So that would be the case for DNA. Let's take a closer look at gel electrophoresis for DNA. So DNA can essentially be cut into fragments using restriction endonuclease. Different DNA samples will generate different fragment lengths. And so the fragments are going to separate through this gel because DNA is negatively charged because of its phosphate group on each nucleotide. Now, DNA samples are placed into this agarose gel, and fragment size is calculated by comparing it against known industry standards. Specific sequences can also be identified by incorporating a complementary radio-labeled hybridization probe, transferring the separated sequences to a membrane and then visualizing through Southern blotting or autoradiography. So if we're taking a look at proteins, we now have a slightly different procedure. Proteins essentially can be folded into a variety of shapes and sizes, and they have positive regions and negative regions as well. There's no clear positive or negative overall net charge. So proteins have to first be treated with an anionic detergent, such as SDS, in order to linearize and impart a uniform negative charge on the protein. The protein samples are then placed into a polyacromide gel, so that's the main difference, the two different types of gels. And that polyacromide gel has pores in it, and afterwards your protein, your results are going to be compared against industry standards to figure out the size of your protein. Now the separated proteins are transferred to a membrane over here, like this. And then target proteins are identified by staining with specific monoclonal antibodies. And that's the process of Western blotting. So that pretty much sums it up for gel electrophoresis. It's how we can take advantage of an electric field and have biological results from it. The next thing I want to talk about is neuronal signaling. So from neuronal signaling, we can also take a look at cell membranes. Now, the anatomy of a neuron is not something I want to focus too much on, but this is what a neuron would look like. You have the axon over here with an axon over here with the cell body and dendrites branching off of the soma or the cell body. And the axon hillock is where the axon starts, and a neuronal signal is sent down into the synaptic terminals, passed on to another cell body or another neuron. So a nerve impulse happens when an action potential reaches the axon terminal, and that's when a voltage-gated calcium channel opens, and the calcium ions are going to enter the terminal, cause a release of neurotransmitters into the synaptic cleft over here, and they diffuse across and activate receptors to pass on the signal. But what's most important here is the fact that calcium is positively charged, and it's going to generate membrane potential, a change in membrane potential as the voltage-gated channels open and close. So taking a closer look at the axon, we do have intracellular and extracellular fluids, and both of these are electrical conductors. We haven't really looked at conductors yet, but essentially it just means that charge can flow through them. And these two intra and extracellular fluids are separated by a membrane. So at resting potential, there's no current and the electric field is zero in these regions. But when a membrane is depolarized, there's going to be an electrical current and a field that's created to push the signal along. So let's take a look at a traveling nerve impulse. You have the soma, axon, and axon terminal, and this is the order or the direction the nerve impulse is going to go along. And at first, you have a depolarization event happening. Afterwards, that depolarization event is going to pass on to the next portion of the axon, and it keeps passing on while the previous portion repolarizes and goes back to a resting membrane potential. So the depolarization is going to spread down the axon because the calcium channels have opened, and afterwards you have the sodium and potassium channels taking turns to open and close for repolarization. So this is not something that I want to go through in much detail. If you want to go through it in detail, Physics 280 and Physics 380 will go through that as well. Now, for the potential difference, microelectrode measurements show that the potential within the cell is about 70 millivolts less than outside. So we can say the resting membrane potential is minus 70 millivolts. Now, we can also calculate the electric field within the membrane, which we assume to be constant just so that we can calculate something. And we approximate the membrane to be 6 nanometers thick. So one of the things we can do is calculate the electric field by looking at a change in voltage divided by a change in distance. So we have minus 70 millivolts divided by 6 nanometers, and we get 1.17 times 10 to the 7 volts per meter. So we have not really learned about electric potential yet, so I will touch on this later, but we are able to calculate the electric field just from knowing the resting membrane potential and the thickness of the membrane. But what I want to focus on now is how we can model the membrane based on what we've learned through Gauss's law. If we take a look at a membrane bilayer, there's going to be two sides to it, almost like a parallel plate. So we can consider one side to be positive and the other side to be negative. And so the electric field distribution would look like this, where you have the electric field going from positive to negative, and you also have some fringe field effects at the edge, where they're kind of just going out. So this is what we call a realistic drawing of what the electric field looks like. In E&M, we really always want to look at an idealized model, so a simplified model. We ignore the fringe fields at the end, and we just take a look at parallel stacks of electric fields going left and right. And you'll also notice these surfaces here. And what we call these are Gaussian surfaces because we want to do some sort of calculation with them to figure out the electric field. So if we focus on one layer at a time, we can actually construct multiple cylindrical Gaussian surfaces where one end lies in a plate and the other end lies outside. So the field is going to be zero within any conductor, so any one of these. So this is 0 and this is 0. And each infinite sheet of charge is going to have a uniform surface charge density sigma. The total enclosed charge must be sigma or rho s times a per sheet, but there are two. So if we actually do the math for and apply Gauss's law, we'll end up with the following equation for the electric field in the middle, which is E equals to sigma over epsilon naught. So try and see if you can derive this yourself, because that is going to be a task for your group in the next assignment.